by Robert Rimac in the RBCs of the chick embryo. Now, amitosis is also called as a direct cell division. It is the only direct cell division. Reason: a direct cell division is a division in which the nuclear membrane remains as such. It does not disappear. If it disappears, it is called as indirect cell division. And second point, there will be a cage type of structure. Today I'll explain that cage type of structure. And then a cage type of structure called a spindle apparatus is not formed in direct cell division and is always formed in the indirect cell division. So the main point is spindle is present or spindle is absent. So a direct during amitosis, amitosis is very much common even in the cartilage cells of human body, in diseased, aged cells of the human body. It is even possible in the endospermic cells of the seeds during development. So when a seed is developing, endosperm cells are also growing in number. So it is possible to see that during endosperm formation, cells may undergo amitosis because their main function is only nutrition. That means cytoplasm. Their nucleus is not so important. Next is meganucleus of Paramecium. Again, same thing, meganucleus controls the functions. And if you remember, I have told you before, Paramecium can have 3 nuclei or 2 nuclei. So the other micronuclei, whether 1 or 2, they are the one which carries the genetic information for the next generation. So during sexual reproduction, a Paramecium's micronucleus plays an important role, but during daily activities and life and food and all, meganucleus controls. Then Cara is a green algae. It has internodal cells. Those cells will also perform amitosis. Then if you talk about bacteria undergoing binary fission, that is also considered, Monneron cell division is also considered as a type of amitosis by some scientists. Now, what happened during amitosis is, if you draw a cell in this manner, nucleus and cytoplasm is there. So it's a very simple process in which first there will be karyokinesis. First the nucleus will start pinching in this manner. And as the pinching of the nucleus is getting over, now the cytoplasm also starts pinching. And finally you will see there are 2 cells formed. So karyokinesis followed by the cytokinesis, but there are many problems with amitosis. So what are the problems? First drawback I have mentioned in the negative points there. There is unequal distribution of chromatin. Chromatin is what DNA wrapped around the proteins. If you remember in the last class also I told you that DNA is negatively charged and the proteins which are required should be positively charged. So those proteins are histone proteins. So the chromatin, the DNA wrapped around the chromatin, wrapped around the histone is called nucleosome, and nucleosomes again wrap around to form a component which is called as chromatin, and then chromatin change into chromosome. This we will be learning in the 9th chapter, and details will come in the 12th class. So the problem with the amitosis is that it's a long piece of DNA, or you can say chromatin. Now a long piece of DNA or chromatin, if it is not undergoing condensation, it can break while the nucleus is undergoing constriction because it's a long thread. It may undergo coiling and cause trouble in the division also. And then because it's a long thread, it is impossible to perform complete duplication or replication of DNA. And before the duplication gets over, unequal distribution of chromatin generally takes place. So it is possible if there are 2 cells getting formed by amitosis, one may get extra DNA, one may get less DNA, or one may get complete amount of DNA, other may get the incomplete amount of DNA. So there are problems with the amitosis. That's why slowly the nature through the course of evolution has developed the better method of division, in which there are maximized chances of equal distribution of the genetic material and least chances of breakage of the genetic material, and that method is called as indirect method of cell division. So direct has many problems, though it's the simplest type of division. It does take place in human body, please remember that. So indirect cell division is a cell division in which the nuclear membrane is going to disintegrate or disappear, and spindle cage or this cage-like structure, spindle apparatus, will develop, which helps in the proper chromosomal distribution, so there will be minimum chances of errors or breakage. Now, indirect cell division, initially mitosis only